everybody. Thank you for joining the webinar today. So I'll start by introducing myself. My name is Jasmine. I'm a user support bioinformatician working at the ENA. So that's the European Nucleotide Archive here at the European Bioinformatics Institute in Cambridge. So originally, I started by supporting data processing for our COVID-19 data portal. So that included sharing the data to partners and also making it easier for researchers to publish their data on our portal. Now my work involves supporting more general data submissions at the ENA, and I deal with everything from sort of small individual submitters who might never have submitted any data to the ENA before, and also organizations who are sharing large volumes of data. Part of my job includes improving and updating the submissions process and making the instructions accessible and available to users. Finally, I also do some more specific work relating to other projects like user outreach, like today, and other jobs for the Pathogens Portal. And the Pathogens Portal is what I'm going to talk about today. And this is the EBI's new tool for supporting and accelerating pathogens research. And it's still very much a new tool, so we do welcome feedback about the portal as well. The purpose of the portal is to provide access to the huge quantity of data stored by us here at the EBI, and it's to encourage open and cooperative data sharing in pathogens research. Here I'll give a summary of everything I'm going to talk about today. First, I will talk about the Pathogens Portal, what it is and the infrastructure behind it. Then I will talk about how data is selected for the Pathogens Portal and a little bit about how we can define a pathogen. Then I'll take you on a tour of the Pathogens Portal, explain some features of the website and how you can use it. I'll also show an example of searching through the Pathogens Portal and explain how you can access the different data types and find your information. And then finally, I'll give you a summary and some contact information for us. So now I'll start with the Pathogens Portal and infrastructure. So this covers what is the Pathogen Portal and what's the infrastructure behind it. So the Pathogens Portal is a website. The URL is given here in the slide, pathogensportal.org. It displays data which is provided from EBI data repositories. The purpose of the portal is to provide the data resources in an easy-to-view format that researchers can use to research pathogenic organisms. A little bit of background about the Pathogens Portal. I'm hoping this picture explains itself, but in case any of you are familiar with the COVID-19 data portal, you will find that a lot of the features of the COVID-19 data portal can also be found in the Pathogens Portal. And this is because the portal is built on an existing visual framework and stack, which we originally used for the COVID-19 data portal. And we've done that because it's a lightweight and predictable way for us to display large amounts of data. And it allows us to display it in a way that's easy for users to navigate. So a little bit of background about ENA in case you haven't heard of it. The ENA is part of the INSDC. The INSDC stands for the International Nucleotide Sequence Database Collaboration. And that's a group of databases which includes the ENA, the NCBI in America, and the DNA Data Bank of Japan. And we work together to sync our data and share biological data around the world in a standardized way. And the ENA is a creator of tools for submission and retrieval, and we exist as the European arm of the INSDC. And the INSDC provides a basis for many other bioinformatics tools that you might have heard of, like UniProt, for example. So what types of data are included in the Pathogens Portal? So currently, we include the European Nucleotide Archive data. So I've introduced you a little bit to the European Nucleotide Archive. It's all nucleotide data. That can be raw read data, assembly data. The Biosamples Archive, which is also technically part of the INSDC, but it's a much wider archive that covers any kind of biosample, which could hold biological data and not just nucleotide data. We also include Europe PMC data. So this is open access publications that can be found and searched for and read via the portal. And finally, I've included the EGA, even though technically the data is not displayed in the Pathogens Portal, and that's because it's not displayed directly because it's private human data that's under controlled access. But we do share some information about the clinical cohort metadata to help you be able to find it. So now I'll talk about how the data is selected for the Pathogens Portal and a little bit about how we can define a pathogen. So there is a huge quantity of data in the ENA. I think last we checked, there was something like 50 petabytes of data. So we're always looking for ways to make this data usable and accessible and findable for the research community. So how do we select which of the ENA data to display in the Pathogens Portal? So there are a lot of organisms that could be a pathogen. And we definitely encourage the pathogen research community to come forward if they have a pathogenic organism that they think should be findable in the Pathogens Portal. Pathogen data is not identified by default or in any computational way, and we continue to add to the list of pathogens that are included. And we identify these using the organism taxonomic ID to display in the portal. So yeah, pathogens, if you ask yourself, what could they be? There's many kinds of pathogens, but what we have narrowed it down to is, first of all, the UK Health and Safety Executive list of biological agents to populate the portal to begin with. So this categorizes pathogens by their containment level, which affects people who are working with them. So we just included all of them in that list to begin with. The WHO, the World Health Organization, also makes a list of priority pathogens, which were already within the Health and Safety Executive list, but we did also use this list to make a subset of pathogens that we consider a priority. And that includes bacterial pathogens showing antimicrobial resistance and some of the most likely viral pathogens to cause a severe outbreak. So we include the priority data. Actually, all viruses are included as pathogens because they are all obligate intracellular parasites. While not all are necessarily pathogenic, this is the best method to ensure that the most comprehensive list of pathogenic viruses are presented in the portal. And that's because viral taxonomy is quite messy. And you can view a list of the pathogens that we have included on the Pathogens Portal, and you can find out how they've been classified into these different groups. Okay, and now a tour of the Pathogens Portal. So here I'll explain the features of the website and how to use it. So when you first land on the homepage of the Pathogens Portal, this is what you'll find. And there's various sections which I'll take you through to begin with. So in the top right, we have some useful pages showing some useful information about the Pathogens Portal. And here, general information is included about the data sources used for the portal. It also provides some information about tools that you can use for sequence data download. And you can also find here guides about how to submit your data to the ENA. In the lower right, you'll find the news feed. So here you can find out about latest developments relating to the pathogen portal. And then there is also the main section of the homepage. So these are shown in the top in the blue text and also on the main page. So you see the same sections in the top and at the center. And there's a number next to each of these data types. I don't know if you can see from the screenshot, but it will tell you how many records are displaying for each data type. So the Outbreak tab holds the WHO priority pathogens data. The Sequences tab, or Pathogen Sequences tab, this holds all of the data, all of the pathogen nucleotide data, and that's reads, assemblies, and analyses. So the difference between the Outbreak tab and the Pathogens Sequences tab, first of all, is that the Outbreak tab is only showing data for WHO priority pathogens. Apart from that, they're quite similar. The Samples tab, here you can browse pathogen sample metadata, so you can search for any kind of information that you think might be linked to a pathogen sample and see what you can find. The Literature tab, here you can browse the latest publications related to pathogens. The Cohorts tab, so as I touched on before about the private clinical data, this Cohorts tab will allow you to browse the pathogen data sets that have been linked with clinical cohort data, including private human data. And then finally, the datahubs. This is a new tool for improving data sharing between groups. I won't cover very much about datahubs today, but you're welcome to ask about it if you're interested. Okay, so I will start talking now in some more detail about finding and viewing sequencing data. So all of the pages have a very similar view. This is a typical layout. This is the view you would get if you clicked on the Outbreak or the Pathogen Sequences button. It's also quite similar for the Samples page. So the Outbreak tab is showing a subset of the Sequences section, just to be clear. And there's some different data types displayed. So these are the data types highlighted here. So we have all, so you can view all of them. There are a lot of records, so you might not want to do that. Sequences, this will show you isolated sequence data, just individual sequences. Analysis, this is additional data types that are not an isolated sequence or a genome assembly. And this includes things like antibiograms, variant call files, and additional other sequence data types that don't fit under the raw read or genome assembly category. Raw reads, so you could select to view only raw read data, and you can filter that by various parameters about read data. The same for any of them, you can filter them. Assemblies, so these are the genome assemblies, and you can view partial or completed genome assemblies from this section. And finally, the studies, which is just another way to search for the sequence data by using the study metadata, which might help you find the information in another way. Yeah, you can use the search bar in the top. You could search for any term, things like strain, lineage, clade, host, assembly software, run instrument, strain, whatever you're interested in, you can type it in there and try it and see what results you get. It's always a good place to start. And then you also have the advanced search section. And we have two types of advanced search here. We have the Portal Advanced Search and the ENA Advanced Search. And these are just two types of advanced search to be able to find the data in a bit more of a specified way. Yes, the left-hand filters. So these allow you to filter the data on the page in a little bit more detail. So this can help you to browse the data, find out how much of a particular record is available on the website. And you can find very quickly how many records are linked to a specific organism of interest, or for a specific country, or by date. And yeah, you can filter by all of these different categories. Center name, collect an institution, if you know exactly what you're looking for. You can also filter by data analysis type. So for the extra type of data that does not fit under sequences or raw reads, you can see what kind of analysis types we hold. Because one of those is AMR antibiograms. So yeah, here the analysis types we have. Some of them are as sequence assemblies stored in a different way. Pathogen analysis, antibiograms, reference alignments, etc. You could filter the read data by the instrument platform. And you could filter the assemblies by if they are full or partial genome. So once you've filtered your results and made your search, you can view the results in the table in the main part of the page. You can edit the visible results table to change what you can see. And that's by using, in the right hand, there's a toggle to show some extra columns and find some extra information. And yeah, so that covers the sequence, that covers the outbreak and pathogen sequences data. So now I'll talk a little bit more about the outbreak tab, because it's not just it's not just showing less data, there's a little bit of extra information shown there. So you can find out the classifications again on the website, see which pathogens have been included in the outbreak tab. And of the total 16 million records in the Pathogens Portal, 2 million of those are outbreak sequences. So on the outbreak tab, we have a next strain reports section, and currently we display next strain reports for monkeypox virus, West Nile virus, and Zika virus. And you can click on the next strain reports and get a nice visualization to find out a bit more information about these particular pathogens. The biosamples, yep, it's similar. You can find it there. There's the table view, and you have the button option available to download your search results. The literature tab, as discussed before, this is from Europe PMC, and you can filter the data using the left-hand side filters by classification, publication, date, the source of the data, and the content if it's open access or preprint, etc. In the cohort browser, you can use the headings to find out what kind of data is included. So the data type will give you an idea of a quick look at what data is available for a particular cohort. For example, sequence data, additional data types, which might be part of the Array Express or Biosamples archive or the EGA archive. And you can use the left-hand menu again to filter by the country, medicine, types of data, etc. So this is quite a difficult filter to show because the clinical metadata is quite tricky to put together into a common and filterable format because of the variability of the data. But you can go on there and have a try and see if you can find anything of interest. We also show a cohorts dashboard, and that will display some more detailed information about the particular cohorts that we have listed in the cohort browser. And this is just an example of one showing the dashboard display, and you can just see how the samples are split by which database they're from, and just read some more information about the study associated. Finally now, I'll talk you through an example of searching for some data on the Pathogens Portal. I don't know if any of you have seen this Simpsons episode, but it's a great one where Homer he discovers a computer for work. But yeah, so I'll talk you through an example of searching for some data on the Pathogens Portal to get you started. And I'll also explain how you can access and download some different data types. Imagine you are searching for some sequencing data. So I've shown an example of the kind of thing that you might be looking for. You might want to find data for Acinetobacter baumannii in Brazil, sampled between the years 2019 and 2022, and compare the same from Paraguay, for example. So if we go to the pathogen sequences section or the outbreak section, you can first pick the data type. So we're going to pick isolated sequences this time. And so you first of all, you pick that. And then you can filter by the scientific name. So you can select the scientific name, left-hand filter, search for your species of interest, tick it. And again, it will show you how many records there are held for that particular data type at that filter level. And then you can put in a date range right down to the day. And that's quite easy to do. And then finally, you can select a country and you can search for your country. And you will always see next to each filter, you will see how many records there are held for each one. So in this case, we found 3 records for Paraguay and 51 records for Brazil for the 3 years between 2019 and 2022. So you can edit the detail to see your results, and you might be able to find more information by editing that table, such as where the samples came from, the host, the study, strain, organism, things like that, subspecies. So if you follow the accession links in the data results, that will take you to the ENA browser. And on there, you can find links between the sequence records and their biosample project records. And that's also where you can find the data files for these. You can also download the results. So you can click the download button in the top left corner, and it will show you this download details box. And you must select the results first. You can only download the visible results at the moment. It's not possible right now to download all results. We're working on improving that, but you can download a small subset, either of the EMBL flat file or the faster format sequences. And you'll get a text file just like that. This is the example of a faster file. You can also download the accession IDs if you want to input the accession IDs into another ENA API, for example, to get the data. And you can download a TSV table containing the metadata. So you can also follow the accession links, to view the results in the ENA browser. And that might help because only the sequence data type is downloadable directly from the Pathogens Portal at the moment, and you would need to find another way to download the raw read data, the assembly data, or the analysis data. So for example, if you wanted to access the genome assembly data, you can follow the accession links and view this individual records on the ENA browser, and you can download them directly from the browser. I've also pasted here the faster file download link, and you could provide the genome assembly accession here to get the download. We do also have tools for downloading assembly data via the ENA bulk downloader tool if you would like to download lots of assemblies in one go. So with the Portal Advanced Search, you can build complex queries and you can access more metadata fields. So with all the searching that I've covered before, you might just be looking around, browsing the data and not really sure what you want to find. But here you can put in some very specific parameters. And if you know exactly what you're looking for, you can include them here and get a much more specific result with everything that you want to find included. And you would just open the Portal Advanced Search and click Add Rule or Add Rule Set to add parameters. And the ENA Advanced Search you can also use. So some of you might be familiar with this before. It existed before the Pathogens Portal, and it is available directly from the ENA browser, in addition to being available on the Pathogens Portal. And we have help documentation published for how to use ENA Advanced Search, but this will allow you to build complex queries, and you can access all of the metadata fields. You can use this as an API to get data downloads, and you can get FTP links to download the data via FTP as well. Thank you for listening. So as a summary, first we covered infrastructure and data inputs to the Pathogens Portal. So I explained a little bit about the INSDC data that comes via the ENA and the biosamples archive, a bit about Europe PMC, and I also mentioned the European Genome Archive, which provides clinical data. I talked a bit about the criteria for inclusion of data in the Pathogens Portal. So that began with the UK Health and Safety Executive List. And we also used the WHO Priority Pathogens List to list our outbreak pathogens, and we also include all virus data because they are obligate pathogens. And I think so anyway. I think most people do. I think there are some. I think someone did find that some are benign, but for the most part, yes. And then finally, the navigation and usage of the Pathogens Portal. So I covered a bit about the outbreak page, the samples, the literature page, the cohort browser, and I explained one quick example of how you can find the data and see how many records there are linked to the data. So I'll provide some contact information here. So there's data submissions help. We have a pathogens data submissions guide findable on the Pathogens Portal. You can also contact our help desk if you're ever struggling with pathogens data submissions. If it takes a long time for response, don't worry, we're getting there. We just get a lot of tickets. You can also contact us. So you can use ENA Path Collabs at ebi.ac.uk as the general contact, or you can contact me directly at jasmine@ebi.ac.uk. And if you don't already, you can follow the ENA on Elon Musk's website, or you can also find us on LinkedIn by searching the European Nucleotide Archive. Thank you very much for taking the time to listen. I'm happy to take any questions.
Thank you, Jasmine, for today's webinar. But there are a few questions in the Q&A box, so I'll read them to you, Jasmine and Nadine. First question is about the data download. Is there any way to download all sequences at once? For example, I'm searching for influenza A virus, and there are more than 250,000 entries, but I can only download maximum of 1,000 sequences each time.
If it's directly from the Pathogens Portal, we do have a limit on the downloads. I'm not sure exactly, but if you use our ENA Advanced Search and get the long list of FTP links from the ENA Advanced Search, you should be able to run a mass download using the FTP download links that you get from the result of the ENA Advanced Search. And also when you do the ENA Advanced Search, you can select which metadata you want to include. And there's a lot of metadata fields that you might not see on the Pathogens Portal, but are included in the ENA Advanced Search API.
Okay, thank you. Would you recommend any tools which are useful for viral metagenomics analysis?
I'm not sure that I can recommend any specific tools for viral metagenomics analysis. We do have the NCBI does have the AMR Finder Plus database where you can view AMR genes that might have been identified by metagenomics analysis. But in terms of specific tools, I don't think we do recommend any particular tool.
How to cite data from Pathogens Portal in publication?
If you are citing data that you've submitted yourself, you can search for this in the ENA help documentation. If you search the word cite, you can find a way to cite the specific study. So usually you would cite the study ID the data is linked to. If it was a search result containing multiple data sets that you then analyzed, you might create your own analysis or data that you submit yourself to the ENA. And if you are using other people's data to create a new analysis, you also have to cite their data when you make your own submission, and then you can cite the ENA study ID. And we do have a guide for including that. in terms of the Pathogens Portal itself, you might be able to cite the portal itself if there was some insight you found just from using the portal on its own, but otherwise it would be through using the study ID.
Okay, thank you. So next question is about the search terms in advanced search. When using the advanced search, can we get a list of all biosamples types that are in there, like when they're using the filters? For instance, if I write rectal swab, and sometimes the terms are written as rectal underscore swab, or there are spelling changes when people are writing fecal FECAL or with FAECAL. So how specific is the match in your search box, and has this been standardized?
So for biosamples search, I think I would have to get back to you on that because I'm not sure exactly how. With biosamples, I believe that it's very specific, but that's only through the biosamples website. Within the Pathogens Portal, I'm not sure exactly if the term is just picked up or if you need to use a specific term. So if you are searching on directly on the Biosamples archive, you do have to use the specific term.
In the search results, some accession entries have missing values like collection dates. So is it missing, or has it not been provided, or are you updating this data time to time?
So if the data is already showing as missing, that means it's missing. So we do have a set of terms where a reason might be given for why the data is missing. So this is the INSDC missing terms list, and there are various reasons that someone can give why the data is missing. For example, it's a lab stock, or it's a control sample, or things like that. So we do have reasons. There are reasons why the data might be missing, and if it shows up as missing on the portal, it's not going to be added. Sometimes we do try and curate and improve the metadata results that show up on the Pathogens Portal, but unfortunately it's not possible to contact everybody to make sure that all the metadata is complete and correct.
How accurate is AMR gene we get from ResFinder or CAR database? How can it be useful in AMR pathogen surveillance?
I think the AMR Finder Plus from the NCBI, that's a tool provided by the NCBI. So I couldn't really say exactly how accurate that information is, but what I do know, it's a relatively popular tool with AMR researchers.
Okay, so we have one new question: how to get clinically relevant pathogens?
So I suppose the most clinically relevant pathogens are probably the WHO priority pathogens. So if you were to visit the Pathogens Portal and visit the outbreak tab of the Pathogens Portal, there you will find the most relevant pathogens. So the WHO identifies the priority pathogens on a range of criteria. So that includes burden to human health, likelihood to be able to resolve. So I think the most clinically relevant ones will probably be the WHO priority pathogens, and you can view what data we have on the ENA about them from the outbreak tab, see how many records there are linked to them, and then we also have some next strain phylogenies on display for some of the viral pathogens.
But also to add, and as Jasmine kind of pointed out earlier in the slides, we're very welcome to feedback on what we include in terms of pathogens, and also in this case clinically relevant pathogens on the Pathogens Portal as well.
So what's the best way for users to provide their feedback if they're using Pathogens Portal?
So I think there is actually a button on the Pathogens Portal on the right-hand side that says feedback that I think is possibly on every page. But you can also email us. So in the submit data section, we have published some email addresses. So we do have some published email addresses on the Pathogens Portal website. And we also have a form on the Pathogens Portal website that you can give any comments or feedback there.